Have you ever wondered how life perpetuates itself? How your cells know to be exactly you? Welcome to the intriguing world of DNA replication. This microscopic marvel, happening in our bodies every second, is the linchpin of life as we know it. But what is it really? In simple terms, DNA replication is the process of making an exact copy of a DNA molecule. You see, every cell in your body contains DNA, the blueprint that dictates everything from your eye color to your height. For life to continue, for cells to divide and grow, these blueprints need to be replicated accurately and efficiently. Imagine you're a master architect, and you've drawn up the plans for a magnificent structure. Now you need to build a replica of this structure in another city. You can't just wing it. You need an exact copy of the original blueprint. That's where DNA replication comes in. Much like that master architect, DNA replication ensures that every new cell gets a perfect copy of the genetic blueprint. This is crucial because without an accurate copy of your DNA, a cell might not function properly. It might not even be you. It's DNA replication that maintains our unique genetic identity, making sure that every cell in our body knows exactly what it's supposed to be. But how does this process actually work? How does a cell take one set of plans and make an exact copy? How does it ensure that not a single detail is missed? It's not as simple as hitting the copy button on a photocopier. No, it's a complex, beautifully choreographed dance of molecules, a process so intricate and precise that it boggles the mind. This, my friends, is the magic and mystery of DNA replication, the silent, ceaseless engine driving the continuity of life. It's the unsung hero of your biology, working tirelessly behind the scenes to keep you, well, you. Intrigued? I thought you might be. Stay tuned to discover the intricate dance of molecules that is DNA replication. To understand DNA replication, we must first understand the structure of DNA itself. DNA, or deoxyribonucleic acid, is the molecule that carries the genetic instructions for life. It is composed of two strands that coil around each other, forming a structure we call the double helix. Each strand is made up of tiny building blocks known as nucleotides, which consist of a sugar, a phosphate, and a base. There are 4 types of bases: adenine, thymine, cytosine, and guanine, often referred to by their first letters A, T, C, and G. A always pairs with T and C with G, creating a perfect match every time. This precise base pairing is crucial in the replication of DNA, ensuring that the genetic information is copied accurately. DNA, with its intricate structure and meticulous base pairing, serves as the blueprint of life, guiding the formation and function of all living organisms. Armed with this knowledge, we are now ready to delve into the process of DNA replication. The first step in DNA replication is akin to unzipping a tightly coiled zipper. But instead of pulling a metal tab down a track of interlocking teeth, we have enzymes doing the heavy lifting in this microscopic world. Let's dive into this process that's happening right now in every cell of your body. Imagine a double helix structure of DNA, coiled and twisted like a spiral staircase. The 2 strands of this staircase are held together by hydrogen bonds, forming pairs of nucleotide bases known as adenine thymine and cytosine guanine. This is the zipper that needs to be unzipped. Enter a special enzyme called helicase. Think of helicase as a tiny molecular motor. It's the one that starts the engine of replication, the one with the job of unzipping the DNA double helix. Helicase attaches itself to the DNA at a specific location, known as the origin of replication. Here, it breaks the hydrogen bonds between the base pairs, effectively unzipping the DNA. As the helicase moves along the DNA, it unwinds the double helix, separating the 2 DNA strands. This creates what's known as the replication fork, a Y-shaped region where the DNA is split into 2 single strands. But there's a catch. The DNA strands are not passive during this process. They are subject to supercoiling, a process where the DNA ahead of the replication fork becomes overwound and the DNA behind it becomes underwound. To prevent this, another set of enzymes, topoisomerases, come into play. They relieve the tension caused by the unzipping, ensuring that the DNA doesn't get tangled up. The unzipping of the DNA double helix is a crucial first step in DNA replication. It's a complex dance of molecular machinery where enzymes work together to take apart the DNA, preparing it for the creation of new strands. A process so intricate, yet so vital, happening billions of times in your body at this very moment. With the DNA strands separated, we are now ready to build new ones. Building new DNA strands is a careful and precise process. Let's dive into the details of this process, known as the elongation phase of DNA replication. If you remember from our previous scene, we've already unzipped the DNA helix. Now we're ready to build new strands. So, how do we go about it? Well, we have a special enzyme for that, and it's called DNA polymerase. This little workhorse of a molecule has a crucial job. It adds new nucleotides to the separated DNA strands, meticulously following the pattern of the original strand. But there's a catch. DNA polymerase can only add nucleotides in one direction, from the 5' end to the 3' end of the new strand. This directional constraint results in the formation of two different types of strands, the leading strand and the lagging strand. The leading strand is the lucky one. It's synthesized continuously, as it runs in the same direction that the DNA helix is unzipping. DNA polymerase just moves along the strand, adding nucleotides as it goes. But the lagging strand, it has a tougher job, because it runs in the opposite direction to the unzipping helix. It's synthesized in fragments, not continuously. These fragments, known as Okazaki fragments, are later stitched together by another enzyme called DNA ligase. Still with me? Great, let's recap. In the elongation phase, we build new DNA strands. DNA polymerase is our main player, adding new nucleotides to the separated strands. We have two types of strands: the leading strand, which is synthesized continuously, and the lagging strand, which is synthesized in fragments. And what's the outcome of all this? The result of this process is two identical DNA molecules, each a precise copy of the original. And that, my friends, is the beauty of DNA replication. We start with one DNA molecule, and we end up with two, ready to be passed on to the next generation of cells. Now that we've unraveled the process, let's contemplate the significance of DNA replication. DNA replication, as we've learned, is more than just a biological process. It's the foundation of life as we know it. Every living organism, from the tiniest bacteria to the largest whale, relies on this intricate process to grow, develop, and pass on their genetic material to the next generation. Consider the miracle of growth. From a single fertilized egg, a complex organism with trillions of cells emerges. Each of these cells carries an identical copy of the DNA present in the original egg. It's through the faithful replication of DNA that such astronomical multiplication and growth become possible. Now, think about the process of inheritance. Every child is a unique blend of their parents, carrying a mix of their genetic material. This is a result of DNA replication during the formation of sperm and egg cells. The genetic information is copied, shuffled, and then passed on, creating a new, unique individual. But DNA replication isn't just about continuity, it's also about diversity. The occasional errors that occur during DNA replication introduce slight variations in the genetic code. While some of these variations may be harmful, others could confer advantages, driving evolution and the diversification of life. However, it's worth noting that not all errors are beneficial. Some errors in DNA replication can lead to mutations that cause diseases, including cancer. That's why cells have developed sophisticated mechanisms to correct these errors and maintain the integrity of our genetic information. So, you see, DNA replication isn't just a process, it's a symphony of life. It's about growth and development, about inheritance and diversity. It's about the delicate balance between continuity and change, between order and chaos. It's the process that enables life to persist, evolve, and flourish. Indeed, DNA replication is a fundamental process that underscores the continuity and diversity of life.